So hi, I'm Shrija Ghosh. I'm a PMRF from IIT Kanpur and I'm taking the problem solving sessions for the NPTEL course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur. Okay. So today we will discuss questions related to the 9th week lecture content which had the lectures related to cell metabolism, different pathways that are involved in the metabolic processes inside the cell, and along with that some content about the xenobiotics. So we will see questions related to these topics, and you can ask questions whenever you have doubt. So we will start now. So starting with the first question, which of the following terms is used to describe the increase in concentration of a chemical compound in the tissue of a living organism with respect to water? Option A, live accumulation factor. Option B, bioconcentration factor. Option C, chemical accumulation factor. And option D, chemical concentration factor. So, anyone wants to answer this question? Which is the term that is used to describe the increase in concentration of a chemical compound in the tissue of a living organism with respect to water? So, the concentration of the compound in the tissue with respect to the concentration of that compound in water. What is the term that is used to describe this?
Is it bioconcentration?
Yes, it is bioconcentration factor. So, the correct answer for this question is bioconcentration factor. So, what is bioconcentration factor? It is basically the concentration of any compound that is a contaminant in present in the tissue of a specific organism at a particular time, divided by the concentration of the same compound present in water. So, this factor is important to understand to understand whether any compound will be bioaccumulated or not. Okay, so it is important to see if the compound which is used, like let's say if there is a synthetic compound which is being used as a pesticide, so that how much it will get incorporated in the biota, in the different species that are around us. So, let's say some compound is released in the environment, and then that compound, since we are also present in the environment, that compound we are also inhaling. So, that compound is getting accumulated in our tissues. So, how fast our tissues are getting accumulating this compound with respect to the concentration of this compound in water. So, if this bioconcentration factor is high, that means the concentration is higher in the biota versus water. That means the compound is a very serious contaminant, it's a very serious pollutant, and it is getting accumulated in all the organisms, and it should be cleared out of the system before it poses some health hazards. Okay, so that is why this factor is important, bioconcentration factor. I hope that's clear. So we'll move to the next question. If you have any doubt, you can either post it on the chat box, or you can ask, you can unmute your mic and ask. So question number 2, which of the following is not a xenobiotic compound? So we have to tell which of the following compounds is not a xenobiotic. Okay, first option is dichloro diphenyl trichloroethane, which is basically DDT. Second option is trinitrotoluene. which is TNT, abbreviated as TNT. Third option is glutathione, and fourth option is fluorine. So which of the following is not a xenobiotic compound? Anyone can answer? So what is a xenobiotic compound? First we have to see. So xenobiotic compound is any compound which is not naturally present in the environment. It is basically a synthetic organic compound. So any compound which is synthesized in the lab, which was not present in nature. Okay, so it's not any plant pigment and it's not derived from any of the natural animals or plant sources. It is actually synthesized inside the laboratories by humans and released in the environment. So those are the synthetic organic compounds. So those are called xenobiotics. Okay, so which of the following is not naturally occurring, basically we have to answer. So DDT, the first option, dichloro-diphenyl trichloroethane, it's a synthetic compound. It's a very common pesticide, DDT. It's a very common pesticide used, and it's a very harmful pollutant for the environment, and it poses many health hazards to the to humans as well. The second option also trinitrotoluene, TNT. It's also not a natural compound. It is a synthesized molecule. So it's a toluene molecule with 3 nitro groups attached, and this compound, it is mainly used in explosives. Third option, glutathione, it is actually a natural compound. So the correct answer for this question is glutathione, which is not a xenobiotic. Why it's not a xenobiotic? Because it is naturally available compound, whereas all the other, even fluorine, it is a synthetic compound. It is synthesized inside the labs. So it's not naturally occurring. We don't get fluorine from any of the natural sources. So this is also a xenobiotic. So all three options are xenobiotic. Only glutathione is not a xenobiotic compound. Okay. So dichloro-diphenyl trichloroethane, that is DDT, it is an insecticide used in agriculture. I have told you it's a pesticide, it's an insecticide. TNT also I have told you that it is chiefly used as an explosive. Glutathione, it's a peptide which is found in plants and animals, and it is a powerful antioxidant produced by the liver to protect the body against free radicals, peroxides and heavy metals. Glutathione also eliminates poisons such as drugs, pollutants from our body. So basically all these pollutants that enter our body, glutathione is one of the molecules that are actually helpful in clearing these pollutants from our body. Okay, so it's actually clearing the xenobiotics out of our body, you can say. Okay, and fluorine, again, it's a xenobiotic compound. It is used to make dyes, plastics and pesticides. Okay, this is clear.
It's okay.
Okay, then we'll move to the next question. Question number 3. If the octanol versus water partition log coefficient value of benzopyrene is 6.06, the compound is practically, whether the compound is hydrophilic or hydrophobic. Now, for any compound, the log coefficient of the octanol water partition, so the log of octanol water partition coefficient determines whether the compound is hydrophilic or hydrophobic. So what is the octanol water partition coefficient? It is nothing but the concentration of a particular compound in octanol versus the concentration of particular compound in water. So if the compound is hydrophilic, That means the concentration will be higher in water than in octanol, because it will dissolve in water more than octanol. Octanol is a organic molecule, so you know that hydrophilic compound, if a compound is hydrophilic, it will get dissolved in water. If a compound is hydrophobic, it won't get dissolved in water, it can get dissolved in some organic, non-polar organic solvents. Okay, so octanol is one such organic solvent. So octanol versus water. So if a compound is hydrophilic, it will dissolve in water, hence the concentration in water will be higher. If the compound is hydrophobic, the concentration in octanol will be higher. Okay, now here we have been given the log of the octanol water partition coefficient as 6.06. So we have to tell whether the compound is hydrophilic or hydrophobic. So did you understand the question? Can anyone answer for this? Whether it will be hydrophilic or hydrophobic. If the log of this octanol water partition coefficient is given as 6.06, that means the compound is more soluble in octanol or more soluble in water, we have to tell. Any idea? So if you see here, so KOW is nothing but the octanol water partition coefficient. So it is given as the concentration of octanol, concentration of the compound in octanol divided by the concentration of the compound in water. Okay, so now log of KOW is given as 6.06. That means KOW will be equal to 10 raised to the power of 6.06. Okay, that means concentration of that compound in octanol is 10 raised to the power of 6.06 times more than the concentration of the compound in water. That means the compound is more soluble in octanol than water. That means the compound is hydrophobic. So the answer for this question would be option B, hydrophobic. Okay. So if you see in this graph why this KOW value is important, depending on this log of octanol water partition coefficient, log of KOW, we can actually determine the log of bioaccumulation factor. There are certain graphs that have been plotted by different research groups, and they have actually derived a correlation between the bioaccumulation factor, that is how much a compound is getting bioaccumulated in the environment, that is how much a compound is getting accumulated inside the organisms, inside the tissues of the organisms, with respect to the KOW. So KOW is a chemical factor. You can determine this KOW value for any compound. You can just take any compound and see the concentration of that compound in octanol versus water. So if it is a hydrophilic compound, the value of KOW will be, log KOW will be very low. If a compound is hydrophobic, the value of log KOW will be very high. Okay. So in that way you can determine KOW, log of KOW, and based on that value you can actually see if a compound is getting accumulated in the environment faster or slower. So if you see here, for this particular compound the log KOW value is 6.06, right? So somewhere here. So if you see, the bioaccumulation factor is very high for such compound. That means this compound is a very serious pollutant if released in the environment. Understood? So this is how we determine the octanol water partition coefficient, and this is how we use this factor to study any particular compound, whether it will be accumulated in the environment or not. Okay, whether it will be accumulated in the tissues of different biota that is present around us or not. Okay. So this is clear. Okay. Then we'll move
And
to
ma'am,
the
what is the range of that
yeah.
coefficient?
For KOW,
If it is 7 or 8 like that, what would be the condition?
there is no particular range. So this data set that is shown in this slide, it's from one particular paper. So similar many data sets can be plotted. So this was one, this plot was actually plotted by seeing the bioaccumulation in a algal species chlorella, and they have just compared the KOW value with the bioaccumulation factor. So they determined the bioconcentration factor, the concentration what we have seen in the first question. So they determined the bioconcentration factor by checking the concentration in the tissue versus the concentration in water, and the KOW value for all the compounds are already known, like it can be determined chemically. So this is also known. So they have just made a correlation between the two, that if the KOW value is high, then the bioaccumulation factor will be high. And it is, you can think it logically also, like if a compound is more hydrophobic, it will remain in your system if it enters your body, because your body has uh, most of the biotic systems are hydrophilic, right? They require water for their metabolism. Okay, we also require water for our metabolism. All the microbes, all the higher organisms require water for their metabolism. So if a compound is very hydrophobic, it will remain in the environment because it won't get solubilized in the water easily, and we are not drinking any organic solvents to clear that compound out of our system. So it will get accumulated in our system, and as we go on in the ecological pyramid, if you go to the higher and higher species, like if you start from a small insect and then go up higher till the human, you will see the accumulation of these compounds will increase over the ecological pyramid. So that is why this is a serious issue, and then this factor is very important. It can easily be chemically like it can easily be derived chemically. So you can use this factor to actually check whether the compound you are synthesizing in the lab is safe to the environment or not. So that is the main main logic behind studying this. There is no limit, like you cannot say if there's any limit to that value, because like for this particular data set they have determined till 6 or 7, but you can go high, like if the compound is more hydrophobic, more and more the compound is hydrophobic, this value will keep increasing. Okay, because the concentration in water will keep decreasing, the concentration in octanol will keep increasing. So that will increase this value. I hope that's clear.
And one more thing, if it is hydrophobic, what would be the
It is hydrophobic. It is
range?
hydrophobic because it is less soluble in water and more soluble in octanol. That means it is hydrophobic. If it is hydrophilic, then the value will be lower. The concentration in octanol will be lower and the concentration in water will be higher. So it will, the overall value of KOW will be less than 1, you can say.
Okay, thank you.
Yeah. Okay. So moving on to the next question. Question number 4. How many intermediates are generated in a glycolysis pathway? So we have to tell how many intermediates are generated in a glycolysis pathway. So glycolysis pathway, what is a glycolysis pathway? It's a central metabolism pathway in our body. In our body and in all the microorganisms, like in most of the aerobic uh, organisms that can survive in presence of oxygen, glycolysis is the central metabolism pathway where glucose is the starting material. So whatever food we are eating, it is ultimately broken down to glucose or similar sugar molecules, right? So those, whatever carbohydrates we are taking, so those glucose molecules, they get converted into pyruvate at the end. So glucose is a 6 carbon molecule, so it gets converted into 3 carbon molecule pyruvate, and from pyruvate then it enters another cycle and further metabolism takes place. So glycolysis is the first pathway where glucose is the main substrate and pyruvate is the final product. Okay, but between this glucose and pyruvate there are certain intermediates that are generated. So we have to tell the number of intermediates that are generated in the glycolysis pathway. Okay, so the correct answer for this question is option D, 10. Okay, so there are total 10 intermediates that are generated in the glycolysis pathway. So we'll see a simple diagrammatic representation. So here you can see this is a very simplified glycolytic pathway that they have shown. So you can see this is the glucose molecule, a 6 carbon molecule. Okay, so from glucose, the first intermediate that forms is glucose 6 phosphate. Okay, so glucose 6 phosphate, that means a phosphate molecule is added over the glucose, and to add this phosphate molecule, obviously we need an enzyme for the reaction to go forward. So hexokinase is an enzyme. So any form of kinase, like any enzyme that has kinase written, you can easily imagine that it transfers phosphate. So it will take phosphate from some substrate and it will transfer the phosphate to another substrate. Okay, so in this case hexokinase is taking phosphate and transferring it to glucose to make it glucose 6 phosphate. Okay, after glucose 6 phosphate, the second intermediate is fructose 6 phosphate, and the reaction is catalyzed by the enzyme phosphoglucose isomerase. So phosphoglucose, because this is a phosphoglucose, glucose 6 phosphate means one phosphate molecule over the glucose. Sorry. So phosphoglucose isomerase. What do you mean by isomers? Isomers means both the compounds have the same number of atoms. So both of these compounds, glucose 6 phosphate and fructose 6 phosphate, if you see, the number of carbon, oxygen, hydrogen and the phosphate molecule are same. Only the structure is rearranged in such a way that it makes a different molecule. Okay, so if you see glucose 6 phosphate, it is converted into fructose 6 phosphate with the help of an enzyme called phosphoglucose isomerase. this is an so the enzyme isomerase, so that means it will make an isomer. So here you can see the number of carbons, the number of oxygen and number of phosphate is same, only the structure is different because it's a different molecule. Okay, so these two are isomers of each other and the enzyme is isomerase. So this is the second intermediate. After fructose 6 phosphate, the next intermediate is fructose 1,6 biphosphate. Okay, so now one more phosphate is added. So right now at the 6th carbon one phosphate is there. Then at the first carbon one more at this OH also one more phosphate is added, making it a biphosphate molecule. Fructose 1,6, 1 and 6 is showing at which carbon the phosphate groups are added, and biphosphate means it has 2 phosphates. Okay. And to catalyze this reaction also, a kinase enzyme is used. So in this case it is phosphofructokinase. So whatever the substrate is, here the substrate is phosphofructose, right? And kinase means again it is taking phosphate from somewhere and transferring it to its substrate. Okay. Then the next reaction is the conversion of this fructose 1,6 bisphosphate or fructose 1,6 biphosphate into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. So if you see this molecule, this is a 6 carbon molecule. Okay, and what is happening is this particular bond between these two OH, this is breaking, and in turn this is one of the 3 carbon molecule that is released, that is the dihydroxyacetone phosphate, and then this is another 3 carbon molecule that is released, which is glyceraldehyde 3 phosphate. Okay, so these are the 4th and the 5th intermediate that forms. This dihydroxyacetone phosphate, it can rapidly convert into glyceraldehyde 3 phosphate because these two again are isomers. Number of carbon, oxygen and phosphates are same in both the compounds. So these two are isomers. So the enzyme that will help in the catalysis of this reaction is an isomerase. So this is triose phosphate isomerase. Triose because it's a 3 carbon molecule, phosphate because it has 1 phosphate molecule, and isomerase because it is doing an isomerization reaction. Okay, this part is clear. So 5 intermediates. After these 5 intermediates, so now you have 2 molecules of glyceraldehyde diphosphate. If you see, one coming from fructose 1,6 biphosphate directly and one due to the conversion of dihydroxyacetone phosphate into glyceraldehyde 3 phosphate. Now these 2 molecules of glyceraldehyde 3 phosphate will get converted into 1,3-phosphoglycerate by the enzyme glyceraldehyde phosphate dehydrogenase. So it is a dehydrogenase, that means dehydrogenation will occur. So H2 will be removed. So if you see, there is 1 phosphate here, and it is, and in while doing this it also adds 1 phosphate group at the end at this particular O. Okay. After this 1,3-phosphoglycerate is converted into 3-phosphoglycerate by the enzyme phosphoglycerate kinase. Now what this kinase is doing, this kinase is not adding a phosphate, it is in turn taking the phosphate out of this substrate and transferring it to another substrate. Okay. Here the kinase was taking the phosphate out of another substrate and adding it to this substrate. Here this kinase is taking the phosphate out of this substrate and adding it to another substrate. So phosphoglycerate kinase. and in turn giving the seventh intermediate, that is 3-phosphoglycerate. This 3-phosphoglycerate undergoes another reaction by the enzyme phosphoglycerate mutase, where again rearrangement of this compound takes place to make 2-phosphoglycerate. So, the phosphate group from the third carbon gets transferred to the second carbon. So, these two are also isomers of each other, and the enzyme is phosphoglycerate mutase. From 2-phospho, so this is the eighth intermediate. From 2-phosphoglycerate, enolase enzyme is there. So, this enolase enzyme, so you know the term ene is used for the double bond. So, this is right now OH group is there, that means it is a alcohol, all. We use the term all as a suffix if OH is present, right, in the as a functional group. So, this OH is removed and a double bond is introduced. So, that means an ene group is introduced. So, we know that double bond is there, then it is denoted by ene, carbene. So, like that. So, ene group is introduced and all group is removed. So, enolase is the enzyme that catalyzes this reaction and makes phosphoenol pyruvate. Okay, because the phosphate group is still there. So, phosphoenol pyruvate, this is the ninth intermediate, and then the final product, that is pyruvate, which is made by pyruvate kinase. So, again this is a kinase, so it takes out the phosphate from this phosphoenol pyruvate and transfers it to another substrate, resulting in the final product pyruvate. Okay, is this clear? This is a very important pathway, so you need to have a very clear idea about this pathway. If you don't understand this pathway, then it will be very difficult for you to follow up the coming questions also. Any doubt in this pathway, you can ask right now. It's clear then?
Ma'am, I have
Yes.
one questions
Yes.
regarding, ma'am, suppose in the exam point of view, they can ask the flow chart of the pathway, like after glucose what's next, and either how many parts, how many names of that. They can give an example of the name. For example, if they can give glucose 6 phosphate, then next is a phosphate 6 phosphate. Is it in that pathway of
Yes.
that?
Yes, so they can ask you like what is the enzyme that catalyzes the reaction from glucose to glucose 6 phosphate. That's why you have
Okay.
to remember, like, you have to see what is the substrate and what is the product. So, if the product has an extra phosphate group from the substrate, like glucose to glucose 6 phosphate, you have to immediately think of a kinase enzyme. Okay, and you can write glucose kinase or hexokinase. Hexo is just a term for any 6 carbon molecule, and glucose kinase is also the correct answer for this. Okay, so it's not very difficult to remember. Here also, if you see fructose 6 phosphate to fructose 1,6 bisphosphate, if I ask you the enzyme, think like
Ma'am,
it
which
has
one is the enzyme in this? Just you can tell me once
these,
again.
the ones that are written over the arrow. Okay,
Okay, that is enzyme.
yeah, that is the enzyme that is catalyzing the reaction. So, this is the reactant, this is the product, basically this is the substrate for this enzyme, and this is the product that is forming. Okay, so, or they can ask like uh, what are the two intermediates that are formed after fructose 1,6 bisphosphate by the action of aldolase. So, aldolase is there, that means one bond breakage is happening. So, from this molecule you will get two molecules. So, you can just, you don't have to like mug these up, you can understand how the structures are changing, and then you can make your own structures also, like write your own products also. So, if you see, this is fructose 1,6 bisphosphate, if you see if this bond is breaking, Then you have this carbon with a OH. So this carbon with a OH. Then you have another carbon. So another carbon molecule. That carbon has one O and attached to another carbon molecule. So this carbon is attached to one O molecule and then another carbon molecule. This carbon is attached to an OPO3 2 minus. So OPO3 2 minus. So like that, you if you see from here only, you can see that the what the product is forming, and it is nothing but it is dihydroxy acetone phosphate. Okay. So similarly, this part is glyceraldehyde diphosphate. So like that, questions can come. So in the further in the next questions also we'll discuss few points about glycolysis. So I think that would clear things up for you.
And I have one doubt.
Yes,
Actually,
yes.
that flow chart. This I little understand, but in this chapter, the last chapter, and I'm literally partially understand some chapters, some specific some topics. Like for example, citric acid cycle. I'm literally not understand what's going on that citric acid cycle.
I have a question related to that also. So at that time I'll show you the whole
Yes, ma'am.
cycle
Two,
and
three
I'll
questions
explain.
I have like in my mind, ki, like why this is be happen, why this is not happen. So like after you
Yeah,
complete the session, just I have give me some time.
I will explain. I will explain. Okay.
Okay, fine.
Okay. So the question was how many intermediates are generated in the glycolysis pathway. So that is 10 intermediates are there. Okay. So glucose, first is glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 bisphosphate, glyceraldehyde 3 phosphate, dihydroxyacetone phosphate, 1 3 phosphoglycerate, 3 phosphoglycerate, 2 phosphoglycerate, phosphoenol pyruvate, and then pyruvate. Okay. So the next question, which of the following is a crucial cofactor for the reactions in the glycolysis pathway? So we have to tell which of the following cofactor is important for glycolysis: Mg2 plus, Cu2 plus, S2 minus, and N3 minus. So the correct answer for this question is option A, Mg2 plus. Why? Because right now you saw the glycolysis pathway, so you had different enzymes: hexokinase, phosphofructokinase, glyceraldehyde phosphate dehydrogenase, and phosphoglycerate kinase. So all these enzymes actually require Mg2 plus for their activity. So what happens? So enzyme is a protein molecule, right? So it can bind to the substrate. So it's a protein molecule which has a catalytic pocket, and you have a substrate that can bind to that enzyme and the reaction can get catalyzed. But there are certain enzymes that require some extra help from other metal ions or other forms of ions. So Mg2, so in this case these enzymes actually require help from the metal ion Mg2 plus for catalyzing the reactions. So if you see here, so hexokinase, phosphofructokinase, phosphofructokinase, then glyceraldehyde phosphate dehydrogenase, and phosphoglycerate kinase. All four enzymes, they require Mg2 plus for their activity, and that's why Mg2 plus is a very crucial cofactor for the whole pathway, because out of the 10 enzymes, 4 enzymes are requiring this ion, this cofactor. So this is a very important cofactor for the whole pathway. Okay, without Mg2 plus the pathway won't proceed further because the enzymes won't be functional. So the next question, question number 6, which of the following reaction yields ADP from ATP in the glycolysis pathway? So we have to tell which of the following reactions yield ADP from ATP in the glycolysis pathway. So we have seen the glycolysis pathway right now. So I've told you that there are some enzymes, some forms of kinases that are taking the phosphate group from some substrate and delivering it to the another substrate. Okay, and some kinase are doing what? They are taking the phosphate from the first substrate and delivering it to the second substrate. Okay, so the second substrate is actually ATP. So some kinase can take ATP and deliver it to the substrate, like glucose. In case of hexokinase, they take the phosphate group from ATP and can deliver it to glucose to make glucose 6 phosphate. Okay, and in some cases we can have the phosphate taken out from the substrates in the glycolysis pathway, like phosphoenol pyruvate. So from phosphoenol pyruvate, that phosphate group will be taken out and delivered to an ADP molecule to give ATP. Okay, so it's both way round. So either ADP can form in a reaction or ATP can form in a reaction, depending on what the other product is. Okay, so right now we have been asked like where the ATP is forming in glycolysis pathway. So you have 1,3-diphosphoglyceric acid to 3-phosphoglyceric acid, fructose 6 phosphate to fructose 1,6-diphosphate, phosphoenol pyruvic acid to pyruvic acid, and 3-phosphoglyceric acid to 2-phosphoglyceric acid. Now you have to see from from ATP, ADP is forming. Okay, that means a phosphate is taken out of the ATP molecule. Understood? Like ATP is adenosine triphosphate. So 3 phosphate molecules are present and 1 phosphate molecule is taken out and added to a substrate to give a diphosphate. Okay, so if you see in the first reaction, from the substrate itself, 1 phosphate is taken out, so it is diphospho molecule, and here it is a monophospho molecule, single phosphate group is there. So that means in from this reaction we cannot yield ADP from ATP, right? Because this reaction already a phosphate group is given out. Okay, we have to see a reaction where a phosphate group is taken. If you see the third reaction, phosphoenol pyruvate to pyruvate acid, in this also phosphate is released, phosphate is removed, only pyruvic acid is remaining in the product. In the fourth reaction also, 3-phosphoglyceric acid to 2-phosphoglyceric acid, there is no change in the number of phosphates. So that means that in this reaction also there is no transfer of phosphate group to another substrate. Only in the reaction fructose 6 phosphate to fructose 1,6-diphosphate, you can see one more phosphate group is taken from another substrate to form the product. So that means this is the reaction where ADP is formed from ATP. So if you see here, this is the step, fructose 6 phosphate to fructose 1,6-diphosphate or biphosphate. So here this phosphofructokinase, it is taking one phosphate molecule out of ATP and transferring it to the fructose 6 phosphate to make it biphosphate molecule, and in turn converting the ATP molecule to ADP molecule. So if you see in this figure here, phosphofructokinase. So this phosphofructokinase, it is taking one phosphate out of the ATP, making it ADP, and transferring that phosphate molecule to this fructose 6 phosphate to make it fructose 1,6-biphosphate. Understood? So that's how one molecule of ADP is generated out of ATP. This is clear?
No, ma'am, please repeat once. Not clear.
So ATP is adenosine triphosphate. Okay. So from this triphosphate means 3 phosphate groups are there. Okay. So from this 3 phosphate, 1 phosphate group is taken by this fructokinase, phosphofructokinase, this enzyme, This enzyme is taking one phosphate molecule out of the ATP and giving it to the fructose 6 phosphate. So in turn what is happening? Now this fructose 6 phosphate, that means it has only one phosphate group. Now it has two phosphate group because one phosphate group is taken from the ATP and transferred to this fructose 6 phosphate to make fructose 1,6 biphosphate. Okay, and since one phosphate is taken out of the ATP, now it is diphosphate, so adenosine diphosphate, so it is ADP now. Do you understand?
Yes.
Is it clear?
Yes.
So same thing is happening here also. If you see, glucose, from glucose 6 phosphate, so glucokinase or hexokinase, what it is doing, it is actually taking one phosphate group out of the ATP and giving it to the glucose molecule to make it glucose 6 phosphate. So it has transferred one phosphate out of the ATP to glucose, and in turn now one phosphate is taken out of ATP, then ATP is now what? It is ADP. From triphosphate it is becoming diphosphate because one phosphate is transferred to glucose to make glucose 6 phosphate. Clear? Similarly, if you see in this reaction what is happening, 1,3 bisphosphoglycerate to 3 phosphoglycerate. Sorry. So this kinase, what it is doing, it is taking one phosphate group out of this glycerate, 1,3 bisphosphoglycerate. Okay, so 1,3 at 1 and 3 position, two phosphate groups are there. So one of the phosphate group is taken from this molecule and given to the ADP molecule to make ATP. So adenosine diphosphate will become adenosine triphosphate, and one molecule of phosphate will be removed from this to make 3 phosphoglycerate. So the one phospho group is transferred to adenosine diphosphate in order to make adenosine triphosphate. Okay. Similarly, here also same thing is happening. From phosphoenol pyruvate, the phosphate group is taken and given to ADP so that it can become ATP and this can become pyruvate. Okay. I hope this is clear now. If it's not clear, you can ask. If it's clear, then I'll move on to the next question. Question number 7. Glycolysis starts with dash and ends with dash. Now we have discussed.
Ma'am,
Yes, yes.
ma'am, if we learn that the flow chart thoroughly, then it's be easy to crack the answers because it's little confusing.
If you can learn, then it's good for you. I would suggest that you understand the flow chart so that it would be easier for you to learn it. So that is like, it's both ways. If you can just without understanding, if you can mug it up, then also it will work for you. But then if you understand what is happening, then it will be easier for you to actually learn this. Okay, so it's your wish. You can choose whichever you want to do.
Thank you.
So glycolysis starts with dash and ends with dash. So now we have discussed glycolysis in detail. So you can say it starts with which molecule? It starts with one molecule of glucose. Okay, and ends with what? So it ends with, can anyone answer it, whether it ends with two molecules of pyruvate or one molecule of pyruvate? So I'll give you a hint. Glucose is a 6 carbon molecule and pyruvate is a 3 carbon molecule. So one molecule of glucose will give,
2.
yes, it will give two molecules of pyruvate. So here in this chart also you can see from one molecule of glucose, 2 molecules of pyruvate are generated, and number of ATP, if you see here, 1 ATP is converted to ADP, here also 1 ATP is converted to ADP. So 2 ATP molecules are used up. Here you see 2 molecules of ATP are generated, here also 2 molecules of ATP are generated. So 4 molecules of ATP are generated and 2 molecules of ATP are used up. So remaining how many molecules of ATP are there? 2 molecules. That means the net ATP generation is 2. Okay. I hope that is clear. So you have 4 molecules of ATP generated and 2 molecules of ATP used up. That means the net generation is 2 molecules of ATP. So basically glycolysis starts with 1 molecule of glucose and ends with 2 molecules of pyruvate and 2 molecules of ATP. Okay. So the answer is option number C. Clear? So we'll move to the next question. Under anaerobic conditions, Saccharomyces species, that is yeast, can convert pyruvate into the following. Option A is lactic acid, option B is ethanol and CO2, option C is acetic acid and CO2, and option D is butyric acid and isopropyl alcohol. So under aerobic conditions, yeast can convert pyruvate into which of the following? The correct answer for this question is option number B, ethanol and CO2. So you have seen till glycolysis right now that from glucose molecules, from glucose molecules, the final product is pyruvate. So what is happening after pyruvate, we have to see. So there are actually 3 fates of pyruvate. So from glucose to pyruvate, it is 10 steps of reaction, which is the glycolysis pathway. Okay. So now, Remeka, the question you were asking, so why the other cycles are there? So from pyruvate, now see, ultimate goal of of our body is to completely degrade the food that we are eating to the minimum possible size. So basically it, how much ever it can break. So if it can break till CO2, it will keep on breaking it till CO2. So till now from glucose we have gotten pyruvate. So from pyruvate there are 3 possible pathways under aerobic condition, that is in presence of oxygen, that is all the human beings and all the animals and plants you see around yourself. This pyruvate is converted into acetyl coenzyme A. This acetyl coenzyme A is then transferred, this acetyl coenzyme A then via the citric acid cycle gets converted into CO2 and H2O. So this is the complete oxidation of glucose under in presence of oxygen.
Ma'am, your voice is breaking.
Okay.
I think no problem with that.
Oh, then
That may be helpful.
Then it might be your connection.
Yeah.
You can maybe disconnect and connect again. So this is basically the complete oxidation of glucose. So from glucose, pyruvate, and then acetyl coenzyme A, and then finally CO2 and H2O. So in presence of oxygen, glucose is completely converted into carbon dioxide and water molecules. So 6 molecules of carbon dioxide. So it is 6 carbon molecules. So 6 molecules of carbon dioxide, 2 here and 4 at the end. So 6 molecules of carbon dioxide are generated. Apart from this, there are two other possibilities in case oxygen is not present. So, under anaerobic conditions, two possibilities are there. The first possibility is the fermentation process that occurs in the yeast species Saccharomyces. So, in yeast, what happens? This pyruvate, since there is no oxygen present, under anaerobic condition, this pyruvate is converted into acetaldehyde, and from acetaldehyde, it is converted into ethanol. Okay. So, whatever you have seen, like you must have heard all the alcoholic beverages, those are fermented products, right? So, that fermentation process actually is carried out using these yeast species. So, these yeast, what they can do, they can convert the glucose into pyruvate using normal glycolysis pathway. After pyruvate, what they can do, they can convert pyruvate into acetaldehyde using pyruvate decarboxylase enzyme. This acetaldehyde can get converted into ethanol by alcohol dehydrogenase, and this ethanol is actually the main compound in the alcoholic beverages. Similarly, like any fruit, if it is kept, it gets fermented. So, all the fermentation process, or if you are familiar with the South Indian food, idli batters or dosa batters, we keep those things for fermentation. So, you would have, if you would have seen, the batter is actually kept with the lid closed. Why do we close the lid? To block the oxygen, so that that fermentation process can take place. So, fermentation is a process which occurs in anaerobic conditions. Okay. And in that process, ethanol and CO2 is generated. So, you can see in the dosa batter, bubbles are coming. So, those bubbles are because of the CO2 generation. Even in bread making, those bubbles, like in the bread, you would have seen certain holes. So, those holes are because of the CO2. So, when the CO2 is present, when the bread is baking, those CO2 molecules make gaps. Since it's a gas, it makes gaps inside the bread loaf. Okay. So, this is actually under anaerobic condition, yeast can convert pyruvate into ethanol and CO2. The second, so the third chance that can happen is, and this also occurs in under anaerobic condition only, but it happens in some other microorganisms and in some other cells. So, you must have seen that if you suddenly do vigorous exercise, you will feel pain in your muscles, right? There will be muscle pain if you suddenly, like one day you start running for 10 kilometers, you will feel muscle pain. Why that muscle pain is there? Actually, what happens when you start running suddenly, your body does not get enough oxygen to convert all the energy that is being you like more energy is being used up than it is generated. Okay. So, in that case, what happens? The body starts converting the pyruvate, since it cannot since it does not have enough oxygen to convert it into acetyl coenzyme A, it converts it into lactate, and those lactate or lactic acid, it gets accumulated around the muscles and it actually causes that pain in the muscles. So, that is another pathway where the pyruvate can get converted into. Okay. So, Remika, you were asking why the citric acid cycle was happening. So, it is happening as a part of the continuous metabolism only. So, from glucose, the pathway comes to pyruvate by glycolysis. After pyruvate, it comes to acetyl coenzyme A in presence of oxygen, and after acetyl coenzyme A, the citric acid cycle goes on for the generation of carbon dioxide and H2O, and in turn ATP molecules are generated that can provide you the energy. So whatever food you are eating is ultimately converted into energy by the whole pathway. Okay, so that is the main purpose of citric acid cycle and the glycolysis pathway. All there are certain other pathways that can convert. So apart from the carbohydrates, or apart from the glucose, you are eating other things also, right? There are fat contents in your food, and you are eating proteins. So proteins are broken down into amino acids, and those amino acids can enter at any stage of this straight line. Or fatty acids are also broken down into acetyl coenzyme A, and then it enters the citric acid cycle. So like that, all the things that you are eating are ultimately converted into energy, okay, in form of ATP. This is clear?
Yes, ma'am.
So moving to the next question, question number 9. What is the efficiency of ATP generation if one molecule of glucose yields 36 ATP? Now, it is they have already given that one molecule of glucose is giving 36 molecules of ATP. Okay, and the delta G value for the complete oxidation of glucose is given, and the delta G value for the ATP is given. So let's see how to calculate this question.
Ma'am, one query. Like for
Yes.
example, you can say this is basically used for the ATP to for the fermentations of any batter or any material,
Yeah.
and they can the same work done by ATP synthesize also, ma'am. The ATP synthesize also do the same thing. They can make our food whatever we can eat it, and they can just make the food particle very smallest and all like this.
Yeah, so the ultimate, yes, you can say that the ultimate goal of, see, why are you eating food? To survive. So to, you
Yes,
are
ma'am.
eating food, and in turn your body is like, body requires energy. You
Yes.
have different organs that needs to function, and it requires energy for that. So though that energy is generated by the ATP molecules, and ATP is ATP is synthesized by metabolism of all these uh, food molecules that you are consuming. So glucose and fatty acids and proteins, so everything is getting finally converted into ATP, and the product that is coming out is CO2, which you are already exhaling out. So you are inhaling oxygen and exhaling CO2 out of your system, right? Clear?
Okay.
The during fermentation process, you are not generating it, you are not generating that amount of ATP. See, you, if you see here, during the fermentation process, you are not entering this cycle. So most of the ATP that is generated is during the citric acid cycle, so that is actually not happening here. So that is why this is actually a less energetic process, you can say, energy synthesizing process, you can say. Okay, this is less efficient process, and that's why there are only few organisms that do this thing. And this pathway is also not generating that amount of ATP, because this is also not a common pathway that is found everywhere. It happens only in specific conditions, when there is a lack of oxygen. Only in that case this pathway will occur. Okay. Okay. So in this question, the efficiency of ATP generation has to be calculated if one molecule of glucose yields 36 molecules of ATP. So let's see how to solve this question. So ATP generated from one molecule of glucose, or 1 mole of glucose, is 36 into the energy that is required to synthesize ATP, that is 7.3 kilocalorie per mole. So 7.3 kilocalorie per mole is the delta G value for ATP. So if you see 36 into 7.3, that is 262.8 kilocalorie per mole glucose. That means 1 mole of glucose can generate this amount of energy, 262.8 kilocalorie. Okay. Now for a complete oxidation of glucose, that is glucose in presence of oxygen is completely converted into 6 molecules of CO2 and 6 molecules of H2O. For this reaction, the delta G value, it's highly negative. That means it is very spontaneous reaction, and the value is minus 686 kilocalorie per mole. Okay, so that means the total energy is 686 kilocalorie per mole, and out of which 262.8 kilocalorie per mole is generated by ATP. So if we have to calculate the percentage of ATP generation, efficiency of ATP generation means percent of ATP from the total energy. So 262.8 kilocalorie per mole divided by 686 kilocalorie per mole into 100. So if you solve this, you will get the percentage of the energy generated by ATP is 38.3. That means efficiency of ATP generation is 38.3%. Clear? So the answer is option B, 38%, approximately 38%. I hope this is clear. So the next question is question number 10. Electrons released by the citric acid cycle move down the electron transport chain in which of the following manner? So we have to tell the sequence of the electrons moving in the electron transport chain. So electron transport chain, it comes after the citric acid cycle. So there are certain electrons that are released from the citric acid cycle, and all those electrons move down the electron transport chain and ultimately resulting in the ATP synthesis. Okay, so we have to see the sequence from which, so there are certain complexes. So I have told you about the membrane structure. So it's a phospholipid bilayer structure. So you have two hydrophilic part with the hydrophobic middle region. So in that membrane there are many complexes, many proteins that are present embedded in that membrane, and those complexes, those proteins actually help the transport of electron from one protein to another in a sequential manner, and in turn creating a proton gradient. So this is the elaborative diagram of the electron transport chain. So if you see, this is a mitochondrial membrane. So these two are the hydrophilic parts, the red ones, and the center part is the hydrophobic core. And these are different proteins that are embedded in the membrane. So the electrons that are released from the citric acid cycle, those electrons are carried by the NADH molecules, and those NADH molecules, they give the electrons to the complex 1. First they give the electrons to the complex 1. These star molecules are the electrons. Okay, so the electrons are transferred to the complex 1. Then from complex 1 these electrons are transferred to the coenzyme Q that is attached over the complex 2. So this coenzyme Q, it takes the electrons from the complex 1 and transfers it to complex 3. In the complex 3, those electrons are then transferred from the complex 3 to cytochrome C, which is again present on the membrane. From cytochrome C, the electrons are transferred to complex 4, and from complex 4 it is transferred to H2O molecules. Basically it is transferred to H2O molecules where so these electrons are transferred to the oxygen molecules actually to form the H2O molecule. So oxygen is the terminal electron acceptor. Okay, That means at the end oxygen is present, that is accepting these electrons that are being flown through all these complexes. So, from NADH it is transferred to complex 1, then coenzyme Q, then complex 3, then cytochrome C, then complex 4, and then finally to the oxygen molecule. Okay. So, this is the sequence of the flow of electrons in a electron transport chain. NADH complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then H2O. Okay. So, the correct answer for this question is option number C. Okay. This is clear? Electron transport chain. So, we will move to the next question. Question number 11.
Ma'am, what is the role of electron transport chain?
Yeah,
Why we do the electron transport
I'll
chain?
tell you. So, so you see this blue protein, it is actually ATP synthase, and it is
What is ATP synthase, ma'am? Like ATP.
I am telling you. Yeah. So ATP synthase is basically the enzyme that is synthesizing the ATP molecules from ADP. So all these electrons that are being transferred from complex 1 to complex 3 to complex 4, and ultimately the production of H2O is happening. In the whole process, if you see these green protons, these are released out of the membrane, creating a proton gradient. The concentration of proton outside is much higher than the concentration of protons towards the inner side. Okay. So, once NADH is transferring electrons to complex 1, simultaneously what is happening, it is pushing the protons out of the membrane system. Okay. Then from complex 2, from complex 1 it is transferred to complex 3 via coenzyme Q. From complex 3, when it is transferred to cytochrome C, again many proton molecules are pushed out of the membrane system. So electron is kind of this electron transfer is kind of facilitating the proton transfer outside the membrane system. Now, as a result, what is happening? The concentration of proton is very high outside than inside, and that will create kind of a proton gradient. So, since the proton concentration is higher outside, so all the proton can be now flushed through this ATP synthase inside the mitochondrial matrix. Okay. And when this hydrogen, these proton molecules are transferred through this ATP synthase, it creates a proton motive force. So it kind of gives a force to the enzyme, which basically it's giving an energy to the enzyme to make ATP molecules from the ADP molecules. Okay. So this process, the addition of phosphate to an ADP molecule, it's a highly energetic process. It requires a lot of energy. To give that energy, all these protons are flushed out of the system first during the electron transport chain, and then all these protons then get flushed together through this ATP synthase to generate that high amount of energy, so that this phosphate group can be added over the ADP to make ATP. Okay. So I have told you uh, from the start, if you see glucose, from glucose pyruvate is generated by glycolysis. After that, from pyruvate it is coming to acetyl coenzyme A and the citric acid cycle. During the citric acid cycle, what is happening? Many NADH and FADH molecules are released. Okay, NADH and FADH2 molecules are released. Those molecules, they are carrying the electrons, and those electrons are transferred to the electron transport chain, which ultimately generates a proton gradient. This proton gradient is actually generating a proton motive force, a type of energy that is helping in the synthesis of ATP molecules at the end. Understood? From glucose till ATP, this is the story how it goes. Okay.
Okay.
Okay. So question number 11, conversion of ATP into ADP and PI is basically the phosphate, inorganic phosphate is written as PI. So conversion of ATP into ADP and a phosphate molecule is an example of what type of reaction? So we have seen the
Phosphorylation.
no. So phosphorylation will be the other way round, conversion of ADP into ATP, because phosphorylation means addition of phosphate. Here removal of phosphate is happening. Okay, so removal of phosphate will happen via hydrolysis reaction. Okay, so the answer is hydrolysis reaction. So if you see, hydrolysis reaction means the H2O molecule, it is actually hydrolyzing the bond, breaking the bond between the two phosphate groups. So this is adenine, ribose sugar and 3 phosphate. So this is ATP molecule, triphosphate with the 3 phosphate groups. One of the phosphate bond has to be broken down in order to make it ADP, right? So this H2O molecule, it is actually this H2O molecule is actually facilitating the hydrolyzation of this phosphate bond in order to make ADP. So it's a ATP hydrolysis reaction. Okay. If now phosphate has to be added to this ADP in turn in order to make ATP, then it will be phosphorylation of ADP to make ATP. Okay. Understood?
Yes, ma'am.
So moving to the next question, question number 12. In eukaryotes, while generating ATP molecule from an ADP molecule, the ATP synthase translocate protons from option A, cytoplasm to mitochondrial matrix, option B, intermembrane space to matrix of mitochondria, C is matrix to intermembrane space of mitochondria, and D is cytoplasm to intermembrane space of mitochondria. So this is basically they are asking, I have shown you the ATP synthase enzyme, right? The blue enzyme in that figure. So they are asking the protons are translocated from what point to what point. So if you remember in the figure, if you remember, this is the mitochondrial matrix, mitochondrial membrane. In the membrane, this enzyme is there, ATP synthase enzyme is there. So the protons are actually transferred from the intermembrane space to the mitochondrial matrix. So the answer is translocation of the protons are happening from the intermembrane space to the matrix of mitochondria in order to synthesize ATP molecule by ATP synthase. Okay? Clear?
Yes, ma'am.
So the next question, question number 13. Which of the following has the lowest E naught value? E naught value is basically the reduction potential. So which of the following has the lowest?
NAD plus and NAD plus.
Yes, NAD plus by NADH. So the options are ubiquinone, oxidized form versus the redox form, reduced form, sorry. Cytochrome C, again oxidized versus the reduced form. NAD plus versus NADH, and O2 versus H2O. So the correct answer is NAD plus versus NADH. So if you see, this is the chart of the electron potential, and if you see NAD plus by NADH, it comes under the negative potential, whereas all the other reactions, cytochrome C and O2 by H2O, and then what was the third option? Ubiquinone. So ubiquinone, all of them come under the positive potential. Okay. So moving to the
Yes,
next
ma'am.
question. Which of the following statement is not correct about the electron transport chain? Which of the following statement is not correct about the electron transport chain? Okay, so we have seen the electron transport chain now. So we should be able to answer this. Coenzyme Q is also known as ubiquinone. Complex 1 is also known as NADH dehydrogenase. Cytochrome C accepts electron from complex 2, and coenzyme Q accepts electron from complex 1 and complex 3 both. So now you see, coenzyme Q is also known as ubiquinone, that is a true statement. Complex 1 is known as NADH dehydrogenase, yes, because you have seen NADH was giving the electrons to complex 1 and in turn it was getting converted into NAD plus. So NADH dehydrogenase can actually take the two hydrogen molecules out of the NADH and make it NAD plus. So it can take two electrons and the two protons. So protons will be moved out of the membrane and two electrons will be transferred to the coenzyme Q. Now cytochrome C. Cytochrome C accepts electrons from which complex? I have shown you the figure. Do you remember from which complex the cytochrome C was accepting electron?
Ma'am, complex 3.
Yes, it was accepting electron from complex 3. So this is not correct about the electron transport chain. So if you see in the electron transport chain clearly, the electrons are transferred from NADH to complex 1 and from FADH to complex 2. Both complex 1 and 2 are giving the electrons to coenzyme Q. So if you see, coenzyme Q accepts electron from complex 1 and 2, this is correct statement. Now coenzyme Q will give the electrons to complex 3. Complex 3 will give the electrons to cytochrome C. So that's why that is the wrong statement. Cytochrome C receives electrons from complex 3, not complex 2. Complex 2 can give electrons only to coenzyme Q. Okay. And then cytochrome C will give the electrons to complex 4. Then complex 4 will give the electrons to oxygen. Clear?
Yes, ma'am.
So the next question, question number 15. Choose the correct equation for the energetics of a microbial growth. Fs plus Fe is equal to R, Fs plus Fe is equal to 0, Fs plus Fe is equal to 1 and Rc plus Ra is equal to R. So which of the equation describes the energetics of a microbial growth? So it's option number C, Fs plus Fe is equal to 1. So what is Fs and Fe? Fs is basically the portion of electron donor used for cell synthesis, and Fe is the portion of electron donor used for energy. So all the electrons that are donated, some of the electron is also used for cell synthesis, because in your body different cells are continuously being made, and some cells are continuously dying. Okay, so for that also energy is required, for that also electrons are required. So during a growth of a particular microbe or during a growth of particular cell, whatever electrons are being donated, all the electrons are not transported in the electron transport chain, and it is not completely used for energy. Okay, some of the electrons that are donated are also used for cell synthesis. So both the portions have to be considered, and since these are the portions, so both of them can should contribute to total of 1, because in like it's like probability. So probability of electrons going for cell synthesis plus probability of going electrons going for energy. If you total all the probability, it should always be 1. The sum of all the probabilities is 1. So Fs plus Fe should be equal to 1. Okay. Okay. The next question, question number 16. The ATP synthase powers the generation of ATP molecules with the help of option A, ionic gradient generated by the movement of ions from inner membrane to matrix of the mitochondria during electron transport. Option B is intercellular movement of phosphate ions during electron transport. Option C is intracellular movement of ATP and ADP molecules. And option D is proton gradient generated by the movement of protons between the matrix and the intermembrane space in the mitochondria during the electron transport.
Ma'am, D option.
Yes. So till now, this should be clear. So till now we have been discussing about the electron transport chain and the ATP synthesis.
Yes, ma'am.
So all the protons are first moved to the intermembrane space, and then from the intermembrane space it is again flushed back to the mitochondrial matrix by ATP synthase, which generates a proton motive force which is required for the ATP synthesis. Okay. So this is very this point should be very clear to all of you. So proton gradient generated by the movement of protons between the matrix and the intermembrane space during in the mitochondria during the electron transport, that is what gives the power to the ATP synthase to generate ATP molecules. Okay.
Yes,
So
ma'am.
again this figure you can see it's a very clear figure. I think it's very clear how the electrons are flowing and how the protons are moving and how the protons are again coming back and in turn generating the energy for ATP synthase to synthesize ATP. Okay. So the next question. Question number 17. Which of the following is a terminal electron acceptor for aerobic reactions? So you have to tell which of the following is a terminal electron acceptor for aerobic reaction. So we have seen the electron transport chain. So what was the terminal electron acceptor there? Which molecule was finally taking all the electrons?
Ma'am, oxygen.
Yes. Oxygen was taking all the electrons and then combining with protons to form H2O. Okay. So option
Yes, ma'am.
A, Fe 3 plus state. Option C, SO4 2 minus, and option D, NO3 minus. All these are actually examples of terminal electron acceptor in case of anaerobic reaction where oxygen is not present. So now for humans you can only see aerobic respiration pathway, but for other microorganisms there are certain bacteria that have the capability of growing in the anaerobic conditions. Okay. So there are certain bacteria that can actually grow without the presence of oxygen. So in those cases they are still taking up some or the other carbon source, so though that carbon source is getting metabolized. So where are those electrons going? So there has to be some other electron acceptor other than oxygen. So in that case Fe, SO4 2 minus or NO3 minus, these are like, there are certain sulfur bacteria, green sulfur bacteria, purple sulfur bacteria. These are the bacteria that have the terminal electron acceptor as the sulfate, and there are other nitrifying bacteria that have the terminal electron acceptor as nitrates. Okay. So in case of aerobic respiration or aerobic reaction, oxygen is the terminal electron acceptor. So if you see, there can be three options: aerobic respiration, anaerobic respiration, and fermentation. So in case of aerobic respiration, the terminal electron acceptor is oxygen. In case of anaerobic respiration, the terminal electron acceptor can be any of the metal ions, iron, manganese, and or it can be sulfate ion or a nitrate ion. In case of fermentation, it is the organic compounds. So you have two types of phosphorylation process for ATP generation. One is called substrate level phosphorylation, that is SLP, and the second is oxidative phosphorylation, that is OP. Okay, the oxidative phosphorylation is the one where the ATP synthase is generating ATP. Okay, so electrons are being transferred via different complexes, and then one of the terminal electron acceptor, either these or this, can take the electrons, and then they can in turn generate a proton gradient, via which the proton motive force will help the ATP synthase to generate ATP. So that is the example of oxidative phosphorylation. Even in case of anaerobic respiration, it is named as oxidative phosphorylation, although oxygen is not the terminal electron acceptor, but all the other sequences are same in the electron transport chain as well as the ATP synthesis process. Substrate level phosphorylation, what is this thing? It is basically, you have seen during glycolysis also, there was like one molecule of glucose was making two molecules of ATP, right? So in the glycolysis pathway, only like from glucose till pyruvate, also two molecules of ATP are generated. So the fermentation process that I have told you, like from glucose to pyruvate, and then after pyruvate it is going to ethanol and carbon dioxide. So in that process also, two molecules of ATP are generated out of one molecule of glucose. So in that case, the terminal electron acceptor is an organic compound, and the same organic compound is the main carbon source also. So this is a very minimal process, you can say. In the complete aerobic respiration, where the terminal electron acceptor is oxygen, both substrate level phosphorylation and oxidative phosphorylation is happening. So in the substrate level phosphorylation, that is basically the ATP generated during the during the glycolysis pathway. So why it is called substrate level phosphorylation? Because I have told you the enzymes that are how the ATP was being generated there. The enzymes, what they were doing, they were taking the phosphate group out of one substrate and transferring it to ADP to form ATP, right? So that's why it is, the phosphorylation is happening at the substrate level. From one substrate, the phosphate group is transferred to another substrate. Okay, so that is substrate level phosphorylation. So from substrate level phosphorylation, two molecules of ATP are generated, and from oxidative phosphorylation, other 36 molecules of ATP are generated. Okay, so in total 38 molecules of ATP are generated from one molecule of glucose in prokaryotes under aerobic respiration in presence of oxygen. So in anaerobic respiration, since the maximum is 38 and minimum is 2, it can be somewhere between 2 to 38. So depending on how many electrons are being donated and how many electrons are being accepted by these electrons, which electron acceptor is present, depending on that, the number of ATP generated during the oxidative phosphorylation will differ, and thus the total number of ATP will differ. Okay. So this whole thing is clear. What is substrate level phosphorylation and oxidative phosphorylation?
Ma'am, in fermentation, why it is not oxidation phosphorylation?
So in fermentation, only after pyruvate it is directly getting converted into uh, ethanol
Carbon
and
dioxide.
carbon dioxide. Okay, so there is
Okay.
no other electrons that are being generated that can be passed to the electron transport chain for generation of uh, ATP molecules by oxidative phosphorylation.
Okay.
Okay, you need electrons for the oxidative phosphorylation at the end, right? So in this only after glycolysis, the there are only two more steps and ethanol and CO2 are generated. So there is no other scope of electrons being released that can go into the electron transport chain and generate ATP. Okay.
Okay.
Yeah. Any other doubt?
No, ma'am.
Then we'll move to the next question. Question number 18. Oxidation of N-octane to acetyl coenzyme A is initially catalyzed by which of the following enzyme? So N-octane, it's a alkane. This alkane is converted into acetyl coenzyme A by which of the following the first enzyme in the pathway they have asked. Okay. So the answer for this question is monooxygenase. So monooxygenase is the first enzyme. So if you see here, sequence of reaction for oxidation of N-octane to acetyl coenzyme A. Okay. So N-octane, it's a alkane. That alkane first by monooxygenase enzyme, it is converted into alcohol, that is octanol. From this alcohol, it is converted into octanol by a dehydrogenation. So two hydrogens are removed and dehydrogenation occurs to form octanol. From octanol, from this aldehyde, octanoic acid, that is a carboxylic acid, it is formed. This octanoic acid is nothing but a fatty acid. Any fatty acids, they can be broken down to acetyl coenzyme A in a stepwise manner by beta oxidation. So it's a fatty acid metabolism process. So all the alkane molecules or alkene molecules that we take up, they are ultimately converted into fatty acid, a carboxylic acid basically, which is then converted into acetyl coenzyme A, and from this acetyl coenzyme A again it can enter the, which cycle it can enter? Which cycle acetyl coenzyme A can enter? I have
Ma'am,
told you.
citric acid.
Yes, citric acid cycle or the tricarboxylic acid cycle, right? So acetyl coenzyme. So even if a microorganism is given an alkane as the carbon source instead of glucose, then also it can metabolize that alkane into the carboxylic acid, and then from that carboxylic acid it can convert it into acetyl coenzyme A, and that acetyl coenzyme A can enter the citric acid cycle where the electrons will be generated. Those electrons will be transferred to the electron transport chain, and then from electron transport chain it will uh, it will help the it will help in the ATP synthesis. So basically whatever carbon source you have, nature has evolved certain ways to metabolize those carbon sources so that energy can be generated for the particular organism. Okay, so if a microorganism grows on an octane, then also it can make its own ATP for survival. Understood?
Yes, ma'am.
So question number 19. The steps of anaerobic degradation of alkene follows which of the following steps? So what are the steps that are involved in the anaerobic degradation of alkene? So right now we discussed alkane. So now you have to see for alkenes what are the steps. So the correct answer for this question is,
Ma'am, can you repeat for alkane?
this is what we have shown for an alkane, right? So an octane is an alkane. It has no double bond or triple bond. So it's a single bond carbon chain for 8 carbons. Like you have 8 carbons and all the carbons are attached to hydrogens with single bond. So this is an alkane. If you remove one of the hydrogen from any of the carbon and add a double bond between 2 carbon, it will become an alkene. So that will be an octene molecule. So for alkane, it is getting converted into alcohol, then aldehyde, then carboxylic acid, and from carboxylic acid it is going to the acetyl coenzyme A. Similarly for alkenes, it will also go to the same path. It will go to, from alkene it will go to alcohol, then aldehyde, then carboxylic acid. So here in this pathway you can understand. So any alkane, any number of carbons, 5, 6, 7, 8, 10, 18, anything. So whatever alkane is there, it will first convert it into alcohol, or whatever alkene is there, that will also be converted into alcohol. After the conversion of alcohol, the alcohol will be converted into aldehyde, and in this case the alcohol is converted into a ketone form. So in case of alkene it is converted into aldehyde, and then from aldehyde it is converted into carboxylic acid. Okay, this carboxylic acid is nothing but a fatty acid. Okay, it's a long chain carbon containing carboxylic acid, which is a fatty acid structure, and this fatty acid is then by a process called beta oxidation is converted into acetyl coenzyme A. So this is acetyl coenzyme A.
Okay,
This is
ma'am.
acetyl coenzyme A, CH3CO SCOA. Okay, so alkenes are converted into alcohol, then aldehyde, then carboxylic acid, and then finally to acetyl coenzyme A, which can enter the citric acid cycle. Okay.
Yes, ma'am.
So coming to the next question, question number 20. Synthetic organic chemicals found in the soil that do not occur naturally are called. This we have discussed in the first question only. These are called
Xenobiotic.
xenobiotics. Yes. So synthetic organic chemicals that are found in the soil and they do not occur naturally. So how are they present in the soil? That's the man-made action. So humans have synthesized these chemical compounds and released in the environment, and since they are not naturally produced, it's difficult to remove them from nature also. So these are called xenobiotics. Okay, xenobiotics are the synthetic organic compounds that are not naturally available. So the next question. So we have discussed glycolysis so far. So I hope you can answer this question. You have to match the enzymes with the particular substrates. Can you answer? Any one of you can answer. Hexokinase, pyruvate kinase, enolase and aldolase.
Ma'am, A.
A. Anyone else wants to answer? Ria is saying A. Do you want to answer? So hexokinase, what will be the substrate for hexokinase in glucose?
No, ma'am, sorry, ma'am. I got confused. I think it's B.
Let's start with one enzyme at a time. So what will
Okay.
be the substrate for hexokinase? Hexokinase is the first enzyme of the glycolysis. So the substrate of hexokinase is, glycolysis starts with which molecule?
Glucose.
Yes. So the first substrate is glucose. So hexokinase, the substrate for hexokinase is glucose. The glucose binds to hexokinase to make glucose 6 phosphate, right?
Yeah.
I've told you kinase means it is transferring phosphate group. So it will take the phosphate group from ATP molecule and give it to the glucose to make glucose 6 phosphate. Okay? Then after glucose 6 phosphate, what happens in glycolysis? Isomerization happens. Okay? So that means from glucose 6 phosphate, fructose 6 phosphate will form. And then fructose 1,6 biphosphate will form. Okay. Now tell me, what will be the substrate for pyruvate kinase? So pyruvate kinase, that means it can take a phosphate out of a molecule and give it to another substrate. Okay, from which molecule?
Phosphoenol pyruvate.
Yes, so it can take the phosphate out of a phosphoenol pyruvate and give it to ADP molecule in turn to make ATP. Okay, so this is the example of substrate level phosphorylation also. So it is taking the phosphate out of one substrate, that is phosphoenol pyruvate, and it is giving the phosphate to another substrate, that is ADP, to generate ATP. Okay, so this is one example of substrate level phosphorylation. And when phosphate group is removed from phosphoenol pyruvate, what do we get? What is the product we get?
Pyruvate.
Yes, phosphate is removed, so we get pyruvate as the end product. That is the last product of the glycolysis pathway. Third enzyme is enolase. So what is the substrate for enolase?
Glucose.
No, the substrate for hexokinase was glucose. What is the substrate for enolase? 2
Ma'am,
phos
2-phosphoglycerate.
hmm. So enolase was doing what? It was changing the alcohol group into a ene group. So 2-phosphoglycerate has a OH. So that OH will be removed and in place a double bond carbon will be added. So that is enolase activity. So from 2-phosphoglycerate, phosphoenol pyruvate is formed by enolase enzyme. Okay. The last enzyme, aldolase. The substrate for aldolase is fructose 1,6 biphosphate. It cleaves the molecule into two molecules. Fructose 1,6 biphosphate is cleaved by this aldolase enzyme into two molecules, one of glyceraldehyde 3 phosphate and the second is dihydroxyacetone phosphate. Okay, so the answer is option D. Hexokinase is glucose, pyruvate kinase is phosphoenol pyruvate, enolase is 2-phosphoglycerate and aldolase is fructose 1,6 biphosphate. Okay, so this is the
Yes, ma'am.
glycolysis pathway. You can revise it again. See, if you keep on reading it again and again, you will understand what is happening. So glucokinase or hexokinase, it will, what it will do, it will transfer phosphate from ATP to
ATP to
glucose.
ADP.
Yes, so ATP to ADP and then glucose to glucose 6 phosphate. These are two simultaneous reactions. So the phosphate group is transferred from ATP to the glucose molecule. Okay. Then from
Okay.
glucose 6 phosphate, you are getting fructose 6 phosphate by isomerase. Isomerase, what it is doing, it is just, so glucose and fructose both have 6 carbon molecules. So both the molecules are isomers of each other. So isomerase is actually helping making fructose 6 phosphate from glucose 6 phosphate. Then after this, phosphofructokinase, again it is transferring 1 phosphate from the ATP molecule to the fructose 6 phosphate to make it a biphosphate molecule. Now it has 2 phosphates attached. One was already there and one again it has taken from ATP, and now one phosphate from ATP is taken, so it will make ADP, and in turn fructose 1,6 biphosphate is producing. From this product, fructose 1,6 biphosphate, aldolase reaction, aldolase enzyme will catalyze the reaction to make two products, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. This dihydroxyacetone phosphate, again by an isomerase, it is converted into glyceraldehyde 3 phosphate. So 2 molecules of glyceraldehyde 3 phosphate are there. From 2 molecules of glyceraldehyde 3 phosphate, now 2 molecules of 1,3 biphosphoglycerate will be formed. This will happen by the enzyme glyceraldehyde 3 phosphate dehydrogenase, GAPDH, GAPDH. Okay. Then from this, 1 phosphate will be removed and transferred to ADP to generate ATP. So this is again a substrate level phosphorylation example. So 2 molecules of 1,3 biphosphoglycerate will make 3 phosphoglycerate. So 1 phosphate is removed, so only 3 phosphoglycerate. That means at the third carbon 1 phosphate molecule is present. Then from 3 phosphoglycerate, with the help of enzyme mutase, 2 phosphoglycerate will form. So that phosphate group at the third carbon will be transferred to the second carbon. Then from 2 phosphoglycerate, phosphoenol pyruvate will be formed by the enzyme enolase. So 2 phosphoglycerate, it has a OH group, that OH will be removed and a double bond carbon will be added. So remember like this, all is removed to make ene. So phosphoenol pyruvate, and then from this phosphate group is again transferred to ADP to make ATP, and in turn phosphoenol pyruvate is getting converted into pyruvate by the enzyme pyruvate kinase, and this is also an example of substrate level phosphorylation. Okay.
Ma'am, is this the 10-step reaction of glycolysis?
Yes, this is the
Okay.
10-step reaction of glycolysis. Okay. So from glucose to pyruvate, it is happening in 10 steps. So 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. 10th product is pyruvate. Okay. Okay, the last question of this session. How many reactions that occur in the TCA cycle transfers electrons from a substrate to an electron accepting enzyme? So we have to tell how many reactions that can that occur in TCA cycle. It can transfer electrons to the electron transport chain basically. So the answer for this question is last option, 4. So there are 4 reactions in TCA cycle. So we will see the TCA cycle once. TCA cycle is basically citric acid cycle. It's another name for the citric acid cycle. So I have told you it starts with acetyl coenzyme A. So in glycolysis you have seen, from glucose we are getting pyruvate. From pyruvate, the
Acetyl coenzyme A.
pyruvate is converted into acetyl coenzyme A. This acetyl coenzyme A is now goes in a citric acid cycle. It enters the citric acid cycle where this acetyl coenzyme A is converted into citrate, then aconitate, then isocitrate, then ketoglutarate, alpha ketoglutarate, then succinyl coenzyme A, succinate, fumarate, malate. oxaloacetate, and then again oxaloacetate is cycled back to citrate, and this cycle keeps on going and it keeps on generating NADH and FADH molecules that can go into your electron transport chain. So, here you see there are 3 dehydrogenase enzymes that can actually take the hydrogen molecules out of the substrate and give to the NAD plus molecule to make NADH. Okay, this NADH is made from NAD plus. Okay, and now this NADH is carrying the electrons and the protons, and now this NADH will go and give the electrons to which complex of electron transport chain? NADH will give the electrons to which complex in the electron transport chain? I have shown the electron transport chain, right? From which complex it was starting?
Ma'am, complex 1.
Yes, so NADH will give the electrons to complex 1. So there are 3 reactions where 3 molecules of NADH will come, and each molecule of NADH will give 2 electrons. So 3 to the 6 electrons are transferred to the complex 1. There is one more dehydrogenase which is not NADH dependent, it is FADH dependent. So it can give the hydrogen to a FAD molecule to make it FADH2. Okay, FAD will take 2 hydrogen to become FADH2. This is also carrying 2 electrons, and it can give the 2 electrons to which complex? It can give the electron to complex 2 of the electron transport chain. Okay, so 3 NADH and 1 FADH2. So 4 reactions basically are contributing to the electron transport chain of the cell. So from citric acid cycle, 4 reactions of citric acid cycle are contributing to the electron transport chain to generate ATP molecules at the end. Okay, this is clear?
Yes, ma'am.
Any doubt in any of the question or in this pathway particular?
No, ma'am.
It was very clear. You explained all the questions very clearly. We understood it well.
Okay.
Thank you.
Okay, thank you. So if there's any no further doubt, we'll end this session now. Okay, thank
Ma'am,
you.
I'm having one doubt.
Yeah, yeah.
Like, ma'am, this much of PYQs is enough for this?
uh,
Ma'am, we have to practice more, like
yeah,
how to
you,
practice.
so what you can do is, you can first, mainly you have to understand. So if you see in like, I'll share these slides also. So in these slides the carbon molecules are marked. Okay, so you can see what is actually happening in each step of the reaction. Even for glycolysis, I've added one other path of glycolysis where the carbon molecules are marked. This, so here also the bonds that are breaking or bonds that are getting phosphorylated, everything is marked. So if you see these,
Ria, please mute your mic. Ria. Then only it will be clear.
yeah, so if you see in these if you see these figure, you, it will be easier for you to see, like this OH is marked. So in the next step, what is happening, a phosphate group is added to this particular OH. Then in the next step, this part is marked. What is happening, the isomerization is happening at this part. So basically instead of joining to this carbon, the O is now connected to this carbon directly, and this carbon is now outside the ring structure and OH is there. Then again at this OH another phosphate is added. Then this bond is breaking. So if you see like that, it would be easier for you to understand the pathway, each step of the pathway, and then the names of each enzyme. So I have told you kinase means transfer of phosphate group. Isomerase means isomerization reaction. So like that you can remember. Dehydrogenase means dehydrogenation is happening. So hydrogen will be removed. So uh, like that you can actually see the cycle. So even in the citric acid cycle, you can see each step. If you see the lecture contents also, ma'am has added one figure for the citric acid cycle where they have just shown the number of carbons. So that also you can refer. That is also a very good figure where you can uh, see from 3 carbon how the molecules are broken down and where at what steps the CO2 is being released, because the purpose of the citric acid cycle is finally to release electrons, carbon dioxide and water molecules. This is the final oxidation step. So from glucose till carbon dioxide and water, how the whole oxidation is happening, that you can see. So like here you can see one H2O is released, here you can see one CO2 is released, here you can see one CO2 is released. So like that, if you see the complete reaction, the complete oxidation reaction, like glucose is what, 6 C6H12O6, right? So that C6H12O6, that means there are 6 carbon molecules. So that 6 carbon molecule should be now converted into 6 carbon dioxide molecules, right? So that you can see where all the 6 carbon dioxide molecules are coming. So like that, if you see, like by breaking down each step, if you will see, it would be very easy for you to understand the basic behind the behind each reaction, and it would be easier for you to remember the pathway also. You don't have to like mug up the pathway without understanding. You can understand it and then try to make it even if you don't know the structures, at least try to remember the names, basic like what is happening after what and the names of the enzymes. That would be enough.
Ma'am, we'll get these PPT.
Yes, yes, it's available in the portal. Like I'll upload, I haven't uploaded this particular one, I'll upload it now, so you can access all the PPTs till now whatever I have presented. All the PPTs are available and all the lecture videos are uploaded on YouTube. So all the links are there in the NPTEL portal. So at the left hand corner, left hand bottom corner, you can see a problem solving session tab. So if you click on it, there will be a link. That link is basically to an Excel sheet, so you can see, and all the links are there in that Excel sheet. Okay.
Okay.
So if there's no other doubt, we can end this session.
Yes, ma'am.
Okay, then thank you so much for attending, and